Hi, my name is Diana Behrens. I'm the founder of Survivor Corps, and welcome to our webinar series. Because we know that you can't always get to the experts, we're here to bring the experts to you. And today's topic is one that I'm very passionate about, and I'm very curious about the latest on convalescent plasma. As many of you know who have followed my journey from the beginning, I was the very first convalescent plasma donor at Columbia University back on April 4th. I believe I had the first antibody test done at Columbia given in the country on March 28th. And I have been a huge proponent of donating plasma. I did my full 8 donations at the New York Blood Center by the end of June. I sadly no longer have enough antibodies to donate, although I just got my T cells tested, and I will be asking our guests about that. But now I am a platelet donor because it turns out I have a very high platelet number. So no matter where you are on the spectrum, there's something that you can donate, and it is National Blood Month. If you visit our website, we will give you loads of ways to sign up to donate. So that part is easy in terms of how to do it, but let's talk about why. There's been a real discrepancy between the data and some of the news we've been seeing. And let's get to the bottom of it with our 3 distinguished guests. Today, we're gonna do really brief intros so we can get right into the meat of the matter. We have Dr. Michael Joyner, who from the Mayo Clinic, who is by trade an anesthesiologist, if I could pronounce that properly, and a physiologist. But more importantly for today's conversation, the principal investigator for the Convalescent Plasma Expanded Access Program, otherwise known as the EAP, that really controlled the, the distribution of plasma from the spring up until the fall. If those of you who were with Survivor Corps before the EAP was brought into, remember our early days when we were, I sometimes refer to it as the Tinder of plasma. And it was, hey, are you out there? Do we have an O positive? I had people offering to fly me around the country. And it was the Mayo Clinic's program. And we have Dr. Camille Van Buskirk here, who's the medical director of the blood donor program at the Mayo Clinic and a pathologist by trade, and really came in and saved the day in terms of plasma. And anyone who was looking for plasma in the spring versus the summer can testify to what a miracle that program was. And we, not last but certainly not least, we have Dr. Nicole Bouvier, who is an infectious disease doctor and an associate professor of medicine and microbiology at Mount Sinai Hospital. And unfortunately, we only have her for the first half hour, so I'm going to throw some more questions to her at the beginning of this. And I think that Dr. Bouvier will be able to give an interesting perspective as to the actual practical application of plasma. She's the one who is actually giving it to people in the hospitals and can give us some stories. I know that many of you out there have donated. I know that many of you, your lives have been saved by plasma. So this is an incredibly important topic, and let's get right down to it. So before we start, we are starting, but let's start with this. Convalescent plasma is nothing new. Beyond it being nothing new, it goes back to the 1890s and its development for use against diphtheria, and has had an incredible history since then that brought us to this moment. So whereas we're talking about a lot of, really cutting edge therapeutics, this is maybe cutting edge in terms of figuring out how to use it and in what amounts, in what timing. But the actual idea of transferring antibodies from one person to another is not new. So Dr. Joyner, can you give us a little bit of historical perspective on this?
Yeah, thanks so much, Diana. And it's great to be here with my colleagues. And I think we all appreciate what Survivor Corps has done. And I think it's also important to note that the folks at Mount Sinai, led by Dr. Bouvier, were some of the real innovators here, and were the first people that at least got national recognition, along with the folks down in Houston, to use convalescent plasma clinically. And it's been very helpful to kind of follow their lead in their publications. I think the thing people have to recognize is that when animals or humans recover from a viral or bacterial infection, frequently they have circulating antibodies in their plasma. Those can be harvested and be given to either prevent or treat disease. This was the main way infectious disease was treated prior to the antibiotics and modern vaccination techniques, and was quite common. If you'd had pneumonia in the 1920s or 1930s, it's very likely you would have gotten serum therapy, and the serum probably would have come from animals that had been infected and had their plasma and really serum harvested for the antibodies. Convalescent plasma, things from human donors, has also been going on almost as long. It was used to treat or prevent measles, polio, all sorts of other things. Interest in this sort of therapy really waned with the onset of antibiotics and better vaccinations around the time of World War II and following. But antibody therapy has been used in almost every other major pandemic, or at least tried. And it continues to have some niche applications in things like respiratory syncytial virus in little babies, um, rabies prophylaxis or treatment, and some other areas. But what I think we're seeing now is widespread use because of your efforts and the efforts of this network. And I think about 400,000 patients have been treated in the United States so far. So that's really the history of it. It's got a long history. It's safe, and there's a lot of evidence, both conceptual and experimental and observational, suggesting it's efficacious, especially if given early or in a prophylactic setting.
So just for those of you who might not be as familiar with convalescent plasma, basically, let's use me as an example. I got COVID. My body did exactly what it was supposed to do. It created antibodies to fight off the virus. It created what was almost what I call an internal hazmat suit, which I can then share with other people because those antibodies live in my plasma, which is the yellow, golden sort of watery substance, the part of your blood, not the red part. So by donating my plasma, and I go in and there's an actual centrifuge sitting next to me that splits out the plasma from the red and white blood cells. So they take the yellow stuff and give me back the red and white stuff. So you don't even need the cookies and juice on the way out, really. And then that plasma has those antibodies and can be transferred into somebody who was not able to mount their own immune defense, or it will fortify whatever immune defense they are producing. So have there been, Dr. Joyner, in that history, are there particular viruses that it has been particularly effective, and are there others for which it has
lots of examples
ineffective?
Lots of examples with measles prophylaxis, some pretty good evidence in the 1918 flu pandemic, excellent evidence against something called Argentine hemorrhagic fever, and again, other sorts of prophylactic use against hepatitis and many other forms of viral infection. So there are many examples when it's used early or used prophylactically. Also been helpful against bacterial pneumonia and spinal bacterial meningitis. Again, this would have been pre-World War II.
That's really interesting. So now we take a jump forward to today, Dr. Bouvier, and you are at Mount Sinai. It was Mount Sinai's plasma program that actually inspired the creation of Survivor Corps. I don't know if you know that. But when I was in isolation with COVID, I received multiple requests that were being emailed around looking for survivors to donate their plasma. And I realized at that time, you're having a hard time finding people who are 14 days symptom free and who could test negative for the virus, which people weren't testing negative for until about 21 days. And it was when I got that same email from about a dozen different people, because I was the only person anyone knew at that point who had COVID. I anticipated that there would become a free market for COVID, for plasma among various research institutions, and that a global crisis was not the best time to have a situation where there was competition, and that free markets work in lots of great ways. But during this moment of collective crisis, we needed coordination, we needed collaboration. And so I started Survivor Corps actually to make sure that survivors didn't become a commodity in that respect. And so you all could spend time doing science and we could go get the donors for you. Because we can't do the science. We leave that to you. So Dr. Bouvier, can you tell us what does it look like in real world application in 2001? What has your experience been like with plasma from the beginning at Mount Sinai when it was being used on a compassionate care basis? And what does that mean through to today with the EUA?
So there's been a few different regulatory changes along the way. So we've sort of had to adjust our protocols accordingly, but we actually started looking for donors very early on, before there were really even enough recovered people in the New York City area who had high enough antibody titers at that point to be able to donate. So at that point, it was, a little bit like looking for a needle in a haystack. But we did set up a pretty robust donor screening program, which at this point, I think has screened over 70,000 people for the presence of antibodies that recognize the SARS-CoV-2 virus, and looking at, looking for the donors who have the highest amounts of those antibodies to get them to donate.
We actually co-hosted an antibody party with Mount Sinai a couple of months ago in an effort to identify super donors, people who are at 2,800 who could, and we found a whole bunch of them. Offer people a titered antibody test and they will be at your door.
Yeah. Now it's much easier to find people who have antibodies, but back in March it wasn't. So, once we found our first donor and got them into the New York Blood Center to donate, we did our first transfusion on March 28th, which was under the compassionate use program that the FDA has, which basically allows doctors to give therapeutic or investigational therapeutics if there's no other available FDA approved treatment for a life-threatening condition. So we started transfusing under that program early April, switched over to the EAP once it was open and ready to roll. Transfused.
That means that, so that the EAP was when the Mayo Clinic's expanded access protocol took over and the management and distribution of the plasma.
Exactly.
And the Tinder stage ended.
Yep. And so, after New York had the sort of explosive epidemic that we had in March, April, and May, things calmed down a lot over the summer. We had, very few cases over the summer, started picking up a bit in the fall. And now we're starting to see many more cases of COVID. So we are, our transfusion program is sort of picked back up again, under the EUA. So yeah, I think we've transfused, I haven't counted in a while, but over 500 patients. We're probably up to about 550, maybe even near 600 at this point.
So just to clarify from a medical aspect, the effect of the emergency use authorization, the EUA that was put out was at the end of August, beginning of September. What that meant was that it can be given as a therapeutic. It doesn't need to be given as a last chance at survival, because in the previous situation over the summer, it was only in the spring, it was only being given to those for whom there was no other option. They were really at death's door. Is that correct?
So the EIND program, which is the compassionate use program, really did not specify how sick the patient needed to be to get plasma. So a lot of places were transfusing people who were at death's door. At Mount Sinai, we were focusing on getting to patients early in disease as quickly as we could, and getting plasma into them as soon as they hit the hospital. So based on the literature from prior infectious diseases where plasma has worked, it has seemed to have its best effect early on when the pathogen is still actively replicating inside of the body. We think that most likely the antibodies in plasma have an antiviral effect. They basically prevent the virus from continuing to replicate until the patient's immune system can sort of catch up and help to get rid of it. And so, we started focusing early on, transfusing people who weren't that sick yet, people who were on oxygen by nasal cannula or needing it with a high flow nasal cannula, but before they got intubated, trying to prevent that from happening. So has been our sort of scientific focus. We have given plasma to very sick people as well, but we were trying to really get people before they sort of got really ill.
And Diana, just to amplify that, I think one of the things that worked really well was the preprint mechanism. So the group at Sinai, led by Dr. Bouvier, really got the information out to the rest of the scientific community. And there was a lot of so-called salvage therapy or last-ditch therapy in early April as part of the EAP. But over the course of April, you really saw that to come down to where the vast majority, say 70 or 80% of the units given by May and June were given to the people in the type of clinical conditions that Dr. Bouvier has described. And I know the American Association of Blood Bankers, the group that Dr. Van Buskirk works with, that they had also been very helpful in promoting the idea that you want to avoid, if at all possible, salvage therapy or salvage efforts and get it to people earlier rather than later.
How early? I mean, how early is early, Dr. Bouvier?
As early as we can, meaning that, it can only be given as an inpatient. So the patient has to be admitted to the hospital. So they have to be sick enough to be in the hospital, obviously. But our goal was to try to get the plasma to them as quickly as possible after they hit the door and we knew that they had COVID. So a lot of that depended on how long people were out of the hospital sick. So, there were people, doing the Tylenol and Gatorade thing at home who, after a week or 2 weeks had progressed to the point where, they were having trouble breathing and showed up then. I think one of the things that's been complicated about this disease is that there is both the sort of active viral pneumonia stage where people have trouble exchanging oxygen and carbon dioxide because their lungs are just full of immune cells that are trying to fight the virus. And then there's this sort of after effect where the immune system has gone like a little overboard and a little haywire and started to damage the lungs. And that also impairs oxygen, carbon dioxide exchange. So the symptoms look very similar, and it's hard to sort of tease out just clinically whether a person is sort of in the early pneumonia stage or whether they're getting into the sort of post-COVID immunopathology stage. And so, early on, we didn't really know, if somebody had been, for how long somebody, they could have been sick for 2 days, but they may have had the virus for 2 weeks and not known it. So it was very difficult to sort of just based on history to figure out who were the sort of most likely candidates to benefit from the antiviral properties of convalescent plasma.
Speaking of which, I mean, the monoclonal antibodies can only be given within the first 72 hours of diagnosis. And I know, obviously, I've heard Dr. Bander say 6 hours from admission at Mount Sinai is the goal. But that presents with, sort of a logical question of people get diagnosed on different days, and it depends how long the lab took to get your test result. Why is it not being based on viral load, for example, as opposed to days from diagnosis? And what are there plans to try to get to non-hospitalized patients? Because, this is obviously the first time in our medical history where we have been using the emergency room as a first line of medical defense, and only Tylenol and Gatorade beforehand. Are you seeing evidence that those people who had plasma infusions did not go on to develop the inflammatory and vascular damage that those of us who relied on Tylenol and Gatorade did.
So I think, everybody's a little bit different in how they respond to this virus. Some people are completely asymptomatic, don't even know they have it. Other people, have it kind of like the flu and get over it and they're fine afterwards. Other people, have very prolonged and very difficult hospital courses where they require mechanical ventilation and other things and have, extreme lung damage. We've seen patients who've had to get, be put on the lung transplant list because their lungs are just so devastated by this disease. So, there is a huge spectrum of what this virus can do to an individual. And we don't exactly know which path any one person is going to go down at the early part of their infection. As to why we don't use viral loads, or the viral titer in the patient, the easy answer is because there's no FDA approved test that measures that in a quick way, a quick actionable way. So it's not, we can't do it without a test that can be used in a clinical laboratory.
Nicole, Dr. Bouvier, that's just a fantastic comment too, because when the history of this whole mess is written, people think about diagnostic testing, but there's just so many issues. And I think Dr. Van Buskirk would agree with just testing at every level. If we'd had adequate testing, people who were seropositive could go to the back of the queue for vaccination, things like that. Early, almost point of care testing to help us figure out who needs to get plasma when, how fast, that sort of thing. Just many, there's just been so many things beyond just isn't enough testing so we can do the contact tracing. There's just so many issues with diagnostic testing. If you've seen every company's got their own sort of antibody assay and so forth. So it's really been kind of a Tower of Babel in a lot of ways. And for people like you on the front lines and Dr. Van Buskirk trying to make sense of it for the rest of us, in terms of getting us the products, it's just been very challenging.
Yeah, I found myself yesterday on Twitter explaining to a fairly prominent geneticist on how to read his antibody results.
Yeah, it's not something that everyone knows.
No, there's nothing clear about it. So if that gives you a sense of things, Dr. Buskirk, Van Buskirk, I've not forgotten about you. I'm getting to it in a moment. But just because we're going to lose Dr. Bouvier in a moment, I just wanted to get a sense on your real world impressions. So we're going to do this in a slightly backwards way. And I'm going to go to Dr. Van Buskirk next and ask about sort of the clinical data, the latest data that's been coming out supporting the effectiveness of the early use of high titer plasma. But Dr. Bouvier, can you give us some real world evidence examples of that? Or,
so I think
maybe it's not. I don't mean that. Sorry,
yeah.
that was not meant to be a leading question.
So I think the way I think as a physician, the way a lot of physicians think is with averages. So we look at a large number of people and we see, do most of these people do better with a treatment, worse with a treatment, in comparison to what else we have to offer them. So overall, convalescent plasma does confer benefits in terms of prevention of more serious illness, prevention of death, prevention of requiring mechanical ventilation, for example. When it is given early and when it has a good antibody amount, I think the data is pretty consistent. And there was just a randomized controlled trial published in New England Journal showing that, if you give people high titer plasma within 3 days of their first symptom, they do much better than people who got placebo. So I think the data have been very, very consistent on that. I think one of the difficult things about a new disease is that we have no idea what to do about it. And in New York, we were sort of on the front lines early. And I can tell you in March, we had no idea how to treat people. We didn't know what, lab matters, what labs should we follow? What tells us if a patient is doing well or doing poorly. Simple things that we do now, like proning, putting people on their bellies to breathe. We didn't know that that helped back then. So a lot of this has been sort of learning to fly the airplane in midair. And when you don't have data, you have to just rely on common sense. I think a lot of what we were doing was trying to understand, what do we know about the virus? What do we know about things that other people have tried that seem to help? And what is the most sensible thing to do given a sort of evidence vacuum, which is what, we were in early on, that is luckily starting to fill in.
So 2 final very practical questions for you before we say goodbye to you. One is, who is the ideal candidate for convalescent plasma? Because we all know someone who's gotten diagnosed in the last few days. And how does that person advocate to get plasma in the Mayo Clinic's expanded access protocol no longer exists? So how does one advocate for it? And if you're not in the emergency room within 72 hours, that seems like a bit of a problem. And the most important question I have for you on behalf of this group is, is there any research that Mount Sinai is doing in coordination with your post-COVID care center as to the possible application of convalescent plasma for people suffering from long-term COVID?
Basically, in terms of the first question, the common sense answer is that what we were doing now is screening people for the presence of antibodies to the virus. If they have antibodies, then it is unlikely that us giving them more from a donor is going to do anything more than their own antibodies are doing, just because the amount of antibodies in a unit of plasma are sort of small compared to what your own immune system is able to make once it's sort of geared up. If people don't have their own antibody response, if they're still relatively early in disease within the first couple of weeks, then we will give plasma. And again, there's no evidence showing that that is the right approach, but from a sort of common sense mechanistic understanding of what we think plasma is doing as an antiviral, it sort of makes sense to concentrate giving antibodies to people who don't yet have their own. And we will look at our data after that to see whether, since we've been able to switch to that, whether, that is a reasonable strategy for assigning plasma or not. We don't know. We're still trying to figure that out. In terms of how to get plasma, if the hospital you're in doesn't have it, every institution has their own policies. I mean, some institutions are participating in clinical trials for convalescent plasma. So the only option that you have there is to either be in the trial or not. Some places, don't think that they can participate in the EUA because of various issues with patient protection or, and
I'm sure that Dr. Van Buskirk will have more to add on this subject from a more global perspective, since Mount Sinai is known for giving it.
Yes.
So in terms of those with long-term COVID, before we lose you,
I think that's something that still needs to be studied.
Is it being studied?
Not to my knowledge yet.
How do we get it studied?
There is a lot of stuff in plasma besides just antibodies, and we don't know what all that other stuff does. There are people, the donors' hormones, their cholesterol, their blood clotting proteins, all kinds of things are in convalescent plasma besides the antibodies. We don't know how those things help, hurt, or nothing. It is possible that some of these other things may have some benefits for either acute or long-term COVID. We just don't know. It's something that does need to be studied before we can sort of say one way or the other.
Well, thank you. So I very much look forward to at least those studies being done so we can find out. And that would be incredible. Thank you so much for joining us, Dr. Bouvier, and I turn back to our other 2 guests now. It was a pleasure having you. So Dr. Van Buskirk, Can you tell us about, I know there's some recent data, specifically this Argentina study. Can you tell us about it? What's the latest?
Well, from what I've heard, it's kind of based on some of the prior studies, and we're all showing the same thing basically, where, the earlier you give, the higher, tighter convalescent plasma, the better the clinical response is going to be in those patients. And so I think that's where, going back to that same comment earlier about how do we know who's had it longer, why can't we do testing? Well, again, it's a matter of practicality. We don't have testing for it. And so it's mainly based on time from admission, obviously, for when the hospitals would have access to treating these patients. So basically, all the studies have shown the same thing, basically. The earlier you give it and the higher the titer of the antibodies, it increases that 30-day mortality rate, meaning increases survivability. It decreases some of the long-standing morbidities of it, but that's it's not as strong as the mortality data. And I think that's really where a lot of it's coming from as far as supporting the use of convalescent plasma. Kind of a quick and dirty version, but it just in summary of all the studies that have come out, there've been maybe one or two that there have been issues with the designs perchance, and that's why it didn't show quite the same effect that all these other multiple studies have shown. And so, of course, in some of the earlier studies that were put out, I know Dr. Joyner, I know the group that you've been involved with, looking at the expanded access program data, there were two safety profiles that were put out, and basically it is a safe and effective therapy. The first couple pieces, it was too early to really be able to collect any data that would be meaningful as far as prediction of efficacy. But in the meantime, there have been multiple papers that have come out demonstrating, again, those same points, that the earlier you give it, it's within that 48, 72-hour time frame that you would want to give convalescent plasma. And the units that you're getting, they are now required to be designated. They are tested. The FDA has told us as blood collectors what tests to use. They're approving other tests that are equivalent to give us the highest titers available. And so
what is considered high titer for this use?
So basically, because it's all the whole goal is looking at the neutralizing antibodies, but that is a test that is required to be done in a very complex, high-level biomedical laboratory. Most blood testing facilities, in fact, none of them have the capability of doing those tests. So what the FDA did quite a while back is they did kind of a bake-off of some of these other serology tests, whether they're IgG or combined IgG and IgM test. And so the one that they settled on eventually as the first test to be used for qualification, I'm not going to go into detail of it, but it is a specific test. If you read the FDA guidance, they'll tell you it's an ortho test. And so they gave us a signal to cutoff ratio that was determined to be high titer or just eligible at that point. And so that was 10. So all those units that were tested as having a signal to cutoff ratio as 10 or above, those were considered to be high titer. And those below that were considered to be low titer. And so once they made that verdict, we'd already been collecting all across the country. There have been thousands of units that have been collected. And so we were required to go back through our remaining inventory and relabel them. Kind of tedious, especially for those facilities who had a really good inventory. Locally, our blood donor center, we try to maintain our support of our internal patients, about 70%. It does vary depending on how busy our clinical teams are with transfusing, whether it's for surgeries or traumas or medical patients who are simply anemic for a variety of reasons. So we're small scale, but we have to do the same thing as all the other large scale folks. And so to me, as a practitioner here, it has its advantages as well as some disadvantages. And so coming through this whole COVID experience with all the very rapid changes that were imposed on us with requirements at work, whether or not in products, how do you treat it? Dr. Joyner contacted me very early on saying, hey, what about this convalescent plasma stuff? And it just so happened that the previous year we had done an experimental treatment of 2 neonates for enteroviral infections. There are no medications or vaccines for it because there's so many serotypes. And so basically what we did is we worked with the infectious disease group and the pediatric practice, and we had the parents to donate. We wanted mom for one patient, but mom was a group group O, and she had very high anti-A blood titers. And so, but the baby was an A, group A. And so we had to go to dad. Dad wasn't sure he was infected, but we thought, well, they all live in the same household. Maybe he just didn't realize he felt badly, and he actually had it. So we collected him. We couldn't measure the antibodies. We had to send it out to the CDC for measurements of those because it's not a standard laboratory test. But we transfused anyway. The first baby, I think they waited a little bit too long, so unfortunately it had not a very good outcome. But the second one actually came through. So, and this was based on research from back in the late 1800s, where they had done it previously.
And Diane, I think what Dr. Van Buskirk, I think that's what's really interesting about this is with the work of the blood bankers, our group, the people at Sinai, that just this network that just sort of emerged, the patient advocacy groups and the donors like yourself. What happened is things that had been done either a long time ago or these sort of one-off boutique examples, niche examples that people were trying, was now able to be done at some scale. And one of the things I think that is going to be a positive of this horrible mess is we're going to learn a lot about how to do big science quickly. So I think we will get better, for quote the next time. And I think that there's going to be a reemergence of this sort of therapy for people with weird forms of infectious disease. And I think that's going to be really, really helpful. And I think one of the areas, it's a little bit like the babies that Dr. Van Buskirk mentioned. One of the areas, and it's too bad Dr. Bouvier is gone, is the folks at Sinai pioneered giving the material to people who have very unusual blood disorders who really can't make antibodies. And the stuff was like, it had a remarkable effect in those individuals, remarkable effect. And there was just another case report this morning of a baby that had a remarkable effect in, kind of one of the bubble babies. And that kid survived and was able to go on and get a bone marrow transplant and problem solved. So I think, again, this is absolutely horrible, but you do learn things in a crisis. And the skill set that people like Dr. Van Buskirk and the blood bankers have developed, I think we're going to all be able to live and fight another day with these sorts of approaches down the road.
That's really helpful. I have so many questions left to go, and we've barely scratched the surface, but I do want to ask you, Dr. Joyner, we've now heard sort of, the medical evidence from you and Dr. Van Buskirk. We heard the real world evidence. Why is plasma getting such a bad rap? On the one hand, I look at Twitter and everyone's, and then at the same time, I'm hearing ads on the news, imploring people to donate.
Well, I think we really have to give Dr. Van Buskirk and the blood bankers a lot of credit, because nationally and the blood banking infrastructure, and again, Camille can tell you more about it, is very interesting. But about 25 or 30,000 units a week are being given in the country right now. I guess I'm a little less concerned about some of the ups and downs and opinions and so on and so forth and the geopolitics of this, but because I think that the physicians out in the real world see some benefit to it, certainly see no harm, and see this as a reasonable shot on goal and are pursuing it. So, right. That to me is really where the rubber hits the road, is the person in an average community hospital able to get the material, use it for their patients, use it in a reasonably coherent way early, as Dr. Van Buskirk and Dr. Bouvier mentioned, and then hopefully get some benefit out of it. And I think we have moved in that direction, and as a result, we've made a lot of progress. So I think it's easy to get caught up in the feeding frenzy about, does it work in left-handed people when the moon is full, and versus focusing, standing back and focusing on the big picture. And I think that's one of the advantages of being old like I am, and having seen different trends come and go, is you can maybe have a little bit more appreciation of the signal to noise and recognize that the signals for efficacy, again, as Dr. Van Buskirk mentioned, are quite strong.
So I think that is the message that we really need to get across to the American people, because the difference between monoclonal antibodies and convalescent plasma, of course, and hyperimmune globulin, which we'll talk about in a moment, is, convalescent plasma we need donors. We need people to roll up their sleeves, just like we do to get the vaccine. Although I do believe that everyone who has who have antibodies and who have an active, a robust T cell response should go to the end of the line, or perhaps only get one dose, if we're talking about a scarcity situation with the vaccine, but that we can talk about another day. So speaking, this South African strain, what's the story with this? And this headline saying antibodies don't work. What are they talking about? Plasma? Are they talking about monoclonal antibody? What are they talking about when these headlines just say antibodies don't work and there are problems with this new variant? I would think that convalescent plasma would be your best bet on a new variant, so long as it was locally sourced. No, isn't this like harvesting? If you have allergies, you want to get honey from your area. I mean, isn't this the same thing? Like artisanal plasma?
Dr. Van Buskirk, you go first.
Yeah, I agree. I mean, when you have a new strain of something come out, especially, like this COVID business, you definitely want to collect from your local environment, because they're the ones that are going to likely be carrying the same strain. And so they're going to make the antibodies against that strain. And so that's where your therapy is coming from. You do not want, some kind of whether it's a hyperimmune globulin or just plain plasma from a completely different country or a different region, And so as far as, you've probably heard also the questions about the efficacy of the vaccines that are coming out against some of these variants. I saw where they had said that the one coming that was from the UK, that subsequently it's hanging around about 8 or so of our states here in the States. The regions that were changed in the genome were not affected as far as having the virus be inactivated or causing the immunity in the recipient of that vaccine. So apparently those portions of the viral genome that are responsive to preventing the infection of a person by that virus, it's not affected whatsoever. At least those were the preliminary reports that I heard about that UK strain that's come to the US. Again, you're not going to really know until you have the science and the time to do the science to really know for sure one way or the other, which brings me back to another very common point that I always emphasize with our donors who call me, hey, I've got antibodies. Do I have to still wear a mask? Or, hey, can I go have dinner somewhere in a restaurant with all these strangers? And my automatic response is, well, first off, no, I wish we could, but we can't, because we don't know what is the absolute required level of antibodies to be considered absolutely immune from getting it again. And if you do have antibodies that you think look pretty high, well, maybe they are, but how long are they going to last? I mean, it's just,
in terms of that, what do you think about this new T cell test available from Adaptive Biotechnologies? I took the first one ever last weekend. I'm waiting on my results. I participated in the trial, and so given their accuracy rates, I'm 99.9% sure I'm going to be positive, even though I don't have my last antibody test in November, late October showed I was down to 160.
Okay.
Pretty low, not high enough to bother to donate anymore.
Yeah.
So, with that, what do you think, where do you think this T cell test will play in determining any sort of potential immunity?
I think as far as, down the road, once it becomes more widely adopted and accepted, because up until this point, whenever anybody thinks about, fixing these diseases or curing them, they automatically think of the humoral immunity. Well, don't forget about the cellular immunity. There's a whole bucket of cells that no one pays any attention to that are very much involved in providing immunity to all of us against any kind of invader, whether it's, a virus or a microorganism of bacterial type or whatever it happens to be. So I think it's just a matter of people recognizing the fact that these T cell tests are of value. And I think then it becomes a matter of educating the users, the potential users of these tests, meaning it has to go to the clinicians, it has to roll out to the potential patients themselves. And of course, all the lab folks, we'd be aware of all this stuff pretty early on. And we also advocate that our clinicians would use them as far as looking for immunity. And so, the T cells, the cellular immunity hangs around a heck of a lot longer than the humoral immunity does. And so that's why I think that's where the value is going to come from. At least if you have remaining T cell immunity, you know that you have some immunity, which should give you a little bit of peace of mind. And you would be able to mount an immune response if you are not able to get any sort of antibodies or vaccine on board quickly that could actually do anything.
Diana, and I think what Dr. Van Buskirk said is so important, because we're in a situation now where you are able to identify these new strains because the sequencing and that sort of capability is so high, and it's going to take a while for the epidemiology and the clinical course of these strains to catch up. So that's the first thing. The second thing is this is about suppressing this however you can. So I really agree. I've been vaccinated. I'm going to get my second dose here in a couple of weeks, but it's not like I'm going to just, quit wearing a mask and things like that, because you don't want to take any risk with this. You want to everybody follow the public health guidelines. We'll get the thing under control, and then we'll be able to do a whole lot of look back studies, look at special populations, and really begin to understand the nuances here, and begin to understand, if you're vaccinated once, are you good to go forever? Are you going to need booster shots down the road? Are you going to need something every year, every 5 years, every 10 years, like the Pneumovax? But all we're like Dr. Bouvier said, we're learning to fly the plane while we're actually flying it. And so I think that's what we've got to wait for, the information to catch up. And people are frustrated by that, but I think the other way to look at it is we've learned more faster about this disease than anything in the history. So I think, we're good to go. And certainly the data that's out there in a limited number of patients and in other forms of coronaviruses suggests that if people are challenged years later with the same bug, they may they may quote get it again, but it's a very mild, they get the sniffles or asymptomatic, which would be terrific, because the thing you want to do is keep people out of the hospital and out of the ICU.
We are seeing a fair number of reports of reinfection on Survivor Corps. It's always hard to tell. I am of the belief that our rates of reinfection are above the number of genetically sequenced cases that the news have reported on, and somewhat lower than the reported cases by people, because there are those who never clear the virus. There are inaccurate tests, all kinds of things can go wrong. What are you seeing in the real world? What are you seeing at Mayo in terms of reinfection? And did those people had they mounted an antibody response? What are you seeing?
As far as the ones that are coming towards me for potential donation, having been infected the second time, I have had no donors come to me having gone through a second round. And again, I think you put some very good points out there. It's about your testing. At the beginning, there were so many, just let's be honest, horrible tests. People were thrown together in their bathtub just trying to get them out there and try and help out, but they just weren't the greatest tests. I think a lot of the true positives, once you got the good tests that got through the emergency EUAs for usage, broad spectrum, that's where you really start seeing more true numbers. An interesting thing is having these donors come in. We had a lot of outside of our regular donor population folks come in, and we had people from a long way away that did not use our Mayo tested, or just tests that were completely different than what we used here. And some of them were pretty fly by night, to be honest with you. And so their results, we kind of had to hem and haw around about whether we would use them or we'd ask them to be tested with ours. But I think that was a lot of the original problem of why you would start to see these quote unquote reinfection rates. Also, depending on when you saw them, you may have been seeing an emergence of a different strain. And maybe it was different enough that the original infection, they didn't mount a very good immune response, if any. I've actually had probably a handful of donors come in that we tested their product they'd already donated, and they did not have antibodies. It's a rare phenomenon. It was usually in our younger folks who did not have severe disease. They were pretty much asymptomatic.
I think the other thing, there's some pretty good data about reinfection from the United Kingdom. I think it was 12 or 13,000 healthcare workers were followed, and about 800 of them were seropositive. And, a little over 10,000 were not seropositive. And then they all had clear-cut exposures to infections. And only 2 of the 800 had any evidence of reinfection. And those people were asymptomatic. And these are people, Diana, who are getting, regular screening and so forth. So I think that's the best data. Now, but again, this is going to be something where if we've said it once, we've said it 10 times on the show, is that we've just got to wait for the testing to catch up. and the epidemiology and the testing to sort of, and the clinical trials to all sort of mesh. Normally these things would have been done in series, but now we're trying to do them in parallel. And we were talking today to Dr. Seal and Mills here. We've had a very exciting email exchange all morning long about some testing opportunities. What's going to happen to people who get vaccinated after they've already recovered? Like, so for example, Diana, if you got vaccinated,
Yeah.
what's it going to do to your antibodies, and so on and so forth? And just working with true professionals to try to sort through, what assays are you going to use? When are you going to sample the blood? All this other stuff. It's great to be able to do this in an urgent but yet more organized way than we were doing things 6 months ago. And really, Dr. Van Buskirk and our colleagues, Dr. Mills and Dr. Thiel, are leading the charge there. And we're able to, try to at least have some prospective insight into this versus a lot of educated guesses.
Absolutely. I'm very eager to get my T-cell results, and I will share them with you. In fact, it'll be the they can only say 6 months because that's how long their study has been. But mine will be an interesting case because mine will be 9.5 months since my positive PCR. So it'll be the longest one that they will have seen if it does show positive. And it'll be interesting to track those people who've had their T cells tested to look for reinfection there.
Correct.
That's where I see a lot of use there, untapped. Can we talk a little bit about hyperimmune globulin? Let me see if I've got this straight. When I'm asked what hyperimmune globulin is, I describe it as, like, imagine being a home chef and you're creating like a huge vat of soup, and you're condensing it down to, you're making like a really concentrated, stock. You're going to make the best chicken stock ever, and you are going to boil that thing down until it is so flavorful, you have no idea. And so you're taking the plasma and you're reducing it. You're taking loads of plasma that has, all different kinds of characteristics, presumably, and also very carefully selected properties, and creating this concentrated potion that could be given either as a therapeutic like monoclonal antibodies, but easier to give, because would it, one, be able to be given as an intramuscular shot? Is that how it's being proposed right now? Or is it also a transfusion? Two, is it, how far away are we from it? And three, could it be used prophylactically?
So I'll start and let Dr. Van, because we've actually been working on that. That's part of what we were talking about earlier today, emailing about.
I was just on this email thread. I feel really
yeah, well, I'm not sure I understand them all either, because a lot of the lab medicine people are really smart. They use big words, and
not for me.
they have, and then one of the Dr. Van Buskirk and I have been working together for a long time, and usually my phone calls to her are something like, "Now, Camille, can you explain this to me?" And she does typically a great job. So, anyways, what happens is that you've described the process well. So things are concentrated. There's about 1.5 grams of antibody per liter of plasma, and with the hyperimmune process, you increase that 5 or 10 fold. So it's really almost a distilled product, and it's really almost all IgG, and which is one of the main subfractions of immunoglobulins. And traditionally, this is what happens when people get a gamma globulin shot to prevent, hepatitis, or if they've been exposed to measles and haven't been vaccinated, that sort of thing. So there are several ways you can do it. One is you can give people an IM shot. It's used frequently now, so-called IVIG, the stuff that is not enriched for COVID-19 antibodies, is used to treat people have various forms of immune deficiency. And they're given it IV every couple of weeks or every couple of months, depending on the disease and so forth. It's also used to treat some strange autoimmune diseases. And so that's really, it's you've hit the nail on the head. And people would like to think about passive immunity for post-exposure prophylaxis, or to use in people that not everybody's going to respond to the vaccine. So even if everybody got vaccinated, there are people who just don't make antibodies, because they've had lymphoma, they've had this, they've had that, there's certain drugs that suppress it and so forth. So we are going to need passive immunity, Diana. What did I miss, Camille?
Actually, I really like the description because, I'm sitting here thinking, okay, so let's say you take your donor pool and you put it in a big vat and then you distill it and you fractionate it off. Yeah, you hit the nail on the head.
Yeah, well, you bring up, last time we chatted too, Diana, you brought up something about food, and you must be quite a cook. And when this is all over, I'm coming to your house to eat. That's all I got to say.
It's okay. I'm first, follow me on the Peloton. That's all I want from you, Mike.
Well, my wife handles all that. All I do is just post my.
I know. I'm going to catch up. You're my inspiration. I'm getting back to it now that I'm 10 months past COVID. I'm back on the bike.
Start slow.
And I've been doing a half hour every other day.
Good.
And I am I'm inspired by your posts. So keep on posting your Peloton workouts. And we have a Survivor Core hashtag for all of your Peloton.
Yeah, okay, I will. I'll put that on there.
And this can actually be a little preview of next week's session, which will be on pacing, on physical therapy and how to get back in shape and how to do it slowly and responsibly, and getting right back into an exercise regimen is not what I advise for COVID.
Diana, what you've done here, to think of today, you had one of the world's leading ID experts. You've got one of the world's leading blood bankers. You got myself, who kind of helped organize the 9-ring circus. You're going to have the physical therapists on. You got the community people on. So I think this is what it's going to take, A, to get over the hump here, and then B, to do the lessons learned so that we do a better job with public health in general. I know one of the things that Camille is really interested in, Dr. Van Buskirk, is thinking about what do we have to do to just, and you mentioned earlier, if you can't donate convalescent plasma, donate what you can. And it is National Blood Month. And I think this is going to help us focus on blood collection more generally and all the things blood products do for us.
Oh, absolutely. And that I just want to also note, January 18th is Martin Luther King Day. It is also the National Day of Service. It is the beginning of the inauguration event, although it will not be political in nature. It is American in nature. It is service. And what I've done as my commitment for that day is I signed up to donate platelets, because turns out I'm a platelet super donor. And Survivor Corps will be organizing a virtual blood drive around the country that day, in conjunction with the Presidential Inaugural Committee and the Red Cross and everyone else. And I invite everyone to sign up on the 18th. Don't make your appointment now before they all get booked up. And we can all FaceTime together, and we can have a virtual blood drive party while we're all doing whatever it is that we have to donate.
Diana, and let's get the platelets are so important, because I know the one thing that keeps our blood banking colleagues up at night is platelets. And if Camille, if you could tell us why platelets are so important for you guys with the storage life and all that other stuff, just give us a quick plug. I think, Diana, you've just hit so many nails on the head here, but the platelets are huge.
That is the truth. Thank you so much for doing that, because it's important that, especially for our convalescent plasma donors, once those antibodies drop, and they're all so willing to help, and that's such a valued commodity, and so we always tell them, well, if you want to donate, how about platelets? Some of them, they may not want to do platelets, or else they'd want to do plasma instead, because they're most comfortable doing plasma. Even though it's the same process, you're still hooked up to the centrifuge, that apheresis equipment with the big thing that stands next to you, and your technician will stand there and kind of watch everything, and you can see the products going back and forth.
I love it. I have to tell you, donating has become a real part of my life.
I'm so glad.
Really done it before this, but after my 8 plasma donations, there is nothing more rewarding, more empowering, especially during a global time of need, during a crisis like this, to be able to give back, to save lives. I mean, the last science class I took was in high school. I'm not going into any ER and saving any lives. I'm not a firefighter. I'm not, there are very few things that I can do to save a life, but by sitting in a chair and scrolling through Facebook or Twitter, whatever it is, for a half hour. Platelets take a couple of hours, actually longer than the others, but plasma whole blood takes a half hour once you're in the chair. It is so easy and it is so gratifying to be able to help in a moment when there's really very little that you can do to help. And I know I've said this before, but we as human beings are hardwired to help. It goes against our nature to be told the best thing that you can do is to literally do nothing. And in many cases right now, that is the truth. The best thing you can do is do nothing. Stay home, wear a mask, don't do anything. Just stay where you are if you have it all, if you can. But the one COVID safe activity that you can do, that anyone can do, is go donate blood, plasma, platelets, whatever it is that you have. You can check your local listings you check our website. I'm not going to bother with the rest of these questions because they are all about how often you can donate and this, that, and the other thing. And all that information is available on our website, survivorcorps.com. Please come there. We are there to answer all of your questions. You can sign up today. And remember, it's not about going once. It's about making it a regular habit. Okay. If you can manage to do other things, you can manage to do this. Thank you so much, Dr. Joyner.
Dan, it's always a great pleasure to visit. Really, it is.
I love having you on, Mike. And thank you for taking my random middle of the night texts and calls with all of my questions and ridiculous.
You try to answer them. They're good questions. But but everybody needs to recognize it's the blood bankers are these are people who are like the interstate highway system for medicine, and they've been the interstate highway system here. None of us can go very fast if we don't have good roads, and they are it.
Absolutely.
That's all I got to say. They are it.
Kirk, what you all ran through the emergency access protocol was nothing less than a true miracle for the world, and it bridged what existed before to what can exist now.
Right.
And one of Survivor Corps' projects coming up over the next year will be to memorialize how plasma worked. On suggestion from Dr. Joyner, we are embarking on a project to memorialize the history of plasma through the COVID crisis, because this will not be the last time that this happens. And a lot of inroads were made, and we need to learn so that we know for the future. And we don't have to start back at square one. So thank you both for everything you are doing for the world. We are tremendously indebted. All of you out there, all of you superheroes, please go to the announcements section of our website and fill out every poll there is right now. Thank you very much. And then go donate your plasma, blood, platelets, whatever you have. Thank you. Be safe.